As we have just seen, the keratinocytes of the epidermis provide the physical first line of defense against pathogens, whilst dermal macrophages attack anything that gets through by eating the invader. These are considered part of the skin's innate or inbuilt immune system. Antimicrobial defense is achieved through the double mechanisms of both the innate and the adaptive immune responses. One of the other key cellular components of innate defense are dendritic cells. These form a dense network across the epidermis and dermis that prevents potential invaders going undetected. Dendritic cells get their name from their distinctive shape, which forms long arms called dendrites that they use to search for invading pathogens and damaged cells. Dendritic cells have functional roles in both innate and adaptive arms of the immune response, and are crucial messengers to the main cells of the adaptive immune response, T cells. In the packed epidermis, Langhans cells are the resident cell population. You can see them here in the diagram as the yellow cells gathering in the top layers of the skin. Langhans cells form close interactions with local keratinocytes. Here, both these cell types keep watch for danger signals from each other and the environment. They also have complex abilities to determine false alarms and friendly microbes that form the skin microbiome, another layer protecting the skin from harmful pathogens. One example of the important work of Langhans cells is in priming or preparing the immune response of the skin against Staphylococcus aureus, the bacterium responsible for MRSA. Lots of research is also being done, including by our group, to understand the role of these cells in diseases such as atopic dermatitis, better known as eczema. Langhans cells also have the ability to migrate and leave the skin. Upon this activation, they acquire highly stimulatory abilities for T cells, part of the adaptive immune response. This explains their role as the main link of the innate and adaptive immune system and are responsible for training T cells. T cells are the major component of the adaptive immune response. This is where the immune system customizes its response to a specific bacterium or virus. These extremely specific responses can all be retained in the memory of the immune system by these T cells. We may think of the blood as the most important source of immune cells, especially T cells, but there are twice as many T cells in the skin as there are in the blood. This goes to show the vital role of the skin in preventing infection and remembering the best responses. Because of their specific roles, T cells are split into many different types. The first distinction is in their CD4 or CD8 identity. This means that cells simply have a protein on their surface called CD4 or CD8. Once a cell is infected by a bacterium or virus, it is the role of CD8 T cells to find and kill these cells. We call this the cytotoxic function of these cells. CD4 positive T cells coordinate immune responses and enable customized responses to different pathogens. They are sometimes called helper T cells because of their role in helping other immune cells. These are split into groups called Th1, Th2, Th17, and Tregs. The T stands for T cell, the H stands for helper, and the number indicates the type of molecules, called cytokines, that they produce. The different cytokines that these cells produce instruct other cells to act in very different and specific ways, depending on the type of pathogen detected. These instructions can be very potent and potentially harmful, so they are kept in check by regulatory cells called Tregs that limit inflammation to only the area of the wound and turn off these instructions when they're no longer needed. T cells are named after the organ that makes them, the thymus. At this point, T cells don't have any specific instructions and migrate to the network of lymph nodes around the body to wait. When an antigen-presenting cell, such as a Langhans cell, encounters a bacterium or virus, it takes a piece, called the antigen, away and uses it to train T cells for efficient and longer-lasting responses. As you just saw there, this does not occur in the skin. Instead, it takes place in the many lymph nodes throughout the body. The dendritic cell will show this piece of bacteria to a nearby T cell. This is called antigen presentation. The interaction between the dendritic cell and the T cell determines the type of T cell response and is determined upon whether the antigen is presented to a CD4 or CD8 protein. Once a T cell has received its instruction, it can migrate to the skin and become a resident cell. This means it will stay in the skin for a very long time and provide a memory of the previous pathogen to prevent any further infections. We used to think that the skin is just an inactive layer just to hold our inside bodies together. But from the first responses of the macrophages eating invaders, to Langerhans cells and dermal dendritic cells acting as sentries to identify the invader, and T cells forming long-term protection, we are discovering that the skin is rich in immune cells ready to protect our bodies from the hostile outside environment. Exciting advances in the last few years, such as transcriptomics and the ability to sequence individual cells, have seen a rapid increase in our knowledge of the cell types associated with the skin, and has vastly improved our understanding of skin biology and its role in health and disease.